Welcome to From the Spectrum podcast. This is a podcast about autism. It is my goal to explain what is autism. I intend to use a mixture of scientific literature, personal experience, and opinion. With opinion, I will explain why I feel the way I do and give examples. I will provide links to various references for each episode. For each episode, we will discuss various aspects of autism. The From the Spectrum podcast will mostly avoid causes of autism, and I will try avoiding the increase of diagnoses of late. This is like playing Tug-of-War with barbed wire, and I don't think I want to travel down that path. For today's episode, we will spend time preparing for a future episode on the balance or imbalance of excitation and inhibition. Excitation is activating, turning cells on, and this can be locally within a specific region of the brain or brain areas, or this can be distal, or activating whole systems, regions, or critical parts of the nervous system. That is excitation. Inhibition is the complete opposite. Inhibition turns cells off, turns systems or regions off, and this can be locally or distal. Inhibition quiets or stops things. So, for today's episode, we will discuss a few known genetic factors associated with autism. We will discuss the implications to our biology that leads us to the deficits in social communication and interaction, the sensory processing problems, and the restricted and repetitive behaviors briefly discussed in the last two episodes. We will cover what is going on upstream. Now, I will have a moment when I touch on a significant region of interest of our environment that is a cause. This is rare, and I will not make it a habit. However, for this episode, we have two objectives. One, learn about the biological aspects, and two, prepare for the excitation inhibition episode. The first item is P10, which is short for phosphate and tensin. P10, P-T-E-N. P10 is a loss of neurons or cells, cause overgrowth and increase excitatory synapses, formation and function. Synapses are how cells connect to each other. A major theme today involves the synapses and the synaptic cleft, the small space in between the cells and how they communicate and connect. More on that in a moment. Further, P10 loss is involved with neuronal hypertrophy, hyperexcitability, seizures, remember the closeness to focal epilepsy, and other autistic-like symptoms. P10 implicates Soma Size, migration, spine density, and dendritic overgrowth. A lot of biology here. For the soma is the cell body, cells and glia cells. The soma is the area dendrites branch off and give the cells multiple paths to communicate and travel. Migration is how cells form and develop. They migrate to common areas for better communication and forms spatial relationships. Just think of it as moving closer to friends and family. Moving closer to what brings you together, the common factors. Spine density helps communication within the cells and the destinations. Certain cells have and need specific amounts of spine. Finally, dendritic overgrowth. One idea is synapses strengthen dendrites and causes them to grow. Think of it as a big tree with many thick branches, and then compare it to other less dense trees. Regardless, the nervous system and how we live and operate as a living organism needs proper sizes of these biological components that help cells operate. Dendritic growth essentially is how we organize and connect ourselves inside with the outside, and vice versa. So, really, we are talking about how cells act, how they act and function when called upon. Are they doing what they are supposed to be doing or not? P10 dysfunction is linked to inhibition abilities and glia cells, and burst firing and action potentials. Again, are the cells acting like they should? And is their vehicle functioning, and are the roads they travel down appropriate? This brings us to some glia cells and myelination. When cells fire, they need to fire efficiently and in the correct direction. Myelin is like turning a gravel road into a highway. The highway acts like to cells' path traveling to the synapses, and to the cleft, and to the connecting synapses. Normally, the cleft, the space in between, is 10 nanometers. You cannot imagine how small this is. There are 1,000 nanometers in 1 micrometer, also called a micron. 1,000 nanometers for 1 micron. The thickness of a sheet of paper is about 80 microns. I wonder if the cleft is implicated with autism. Maybe in some parts, like those hyperconnected areas. Maybe they are smaller and larger. I don't know. I'm just thinking if this is something involved with autism. With P10, scientific literature offers hope. The hope is rapamycin, sometimes called mTOR, which means mammalian target of rapamycin. A side note, if you pull up research on aging or longevity, rapamycin is likely involved. Rapamycin and NAD are extremely popular regions of interest for aging and longevity. Okay, so what is it doing for autism? A complex of rapamycin, and don't worry about the specifics with each complex, just the function of it. A complex of rapamycin, the data show prevention of increased Soma size, proper migration, and spine density, and dendritic growth. mTOR regulates cellular health, whereby anabolic cell growth and catabolic cell death. This is why it is so crucial in the aging and longevity literature. Senescence always wins. In other words, senescence, how we age and break down, is undefeated. Next is 16p11.2. Think of a deletion in our biology. Deletion and duplication are common with the genetic functioning. This is an implication to our serotonin. Serotonin comes from the Raphe. The Raphe is at the brainstem and responsible for the serotonergic release in the brain. The dorsal Raphe connects to the ventral striatum. They communicate. Remember the discussion on the striatum, a region of interest for us. Specifically, in the ventral striatum is the Nucleus accumbens. The Nucleus accumbens is one of the most fascinating areas of the brain and body. For a brain region, it is large, and it has two parts, a shell and a core. There are serotonergic receptors in both. The Nucleus accumbens is related to motivation, emotional processing, learning. It has significant amount of NMDA receptors that are crucial for learning. It is also involved in reward prediction, pleasure, addiction, and analgesia, or endogenous pain response. Now, essentially, everything named here can be drawn back to autism. A crucial part of the Nucleus accumbens is socialness. A future episode will dive into the roles of oxytocin and vasopressin and socialness. Specifically, autism versus non-autism, extroverts to introverts, and interesting, female autistics and male autistics, and reviewing some work by the GENDAR Consortium. However, for the sake of today's discussion, the serotonin system from Raphe to Nucleus accumbens and the implications to socialness is an interest. Likely, in the near future, cell-specific targets of serotonin and the Nucleus accumbens could rescue social deficits. If decided by those with social implications caused by autism or any other social disorder or problem, if and when this becomes an option, you have to make that decision. See the links below from Stanford. I want to do a hard stop here, a very hard stop, because it needs to be conveyed. It needs to be known. I will not make a habit of this, but serotonin is synthesized from tryptophan. Tryptophan is an aromatic amino acid, which is a type of protein, a very special type of protein. This is critical and not well known information. Tryptophan makes serotonin and melatonin. Okay, so what's the big deal? The big deal is, as you know, autism came around starting in the late 1930s. Tryptophan, being an aromatic amino acid, activates for UVB light wavelength, specifically around 200 nanometer light. That's within the UVB spectra. Over the past 130 years or so, humans are responding to the shift in the light sources. This is true, and we are poorly responding to it. This is true too. Proteins and gene expression and various aspects of our biology responds differently and requires different types of light. This is true. For instance, do you know anything about melanopsin? Autism is a downstream cause of the shift from sunlight source to the artificial light environment that suffocates us today, and the circadian rhythm mismatch. We have the ability now to extend the day, to see light after dark. Human biology has not evolved for that type of environment. You can look up any health trend and notice that we are trending in bad directions. Few, if any, of our health measurements are doing good. Many things are on the rise, like chronic diseases, Alzheimer's, diabetes, autism, and so forth. All of these are new in the medical literature. Know this. Know this about light sources and our biology and the various functions of the photoreceptors across our body. The brain and skin are connected extremely upstream, much farther upstream than the genetic implications that we are discussing here. The brain and skin, or Neuroectoderm. You won't hear that. You won't hear that any place else, essentially. Aromatic amino acids activate all three of them for UVB and fluorescent for UVA, roughly 353 nanometers. You won't hear this on any other podcast about autism, or any university or research center. Nothing involved with the centralized paradigm. But if you do, please reach out to me and give me their information. Discussions like this, we need those discussions so we can understand what is going on recently with our health. I will provide a link and a warning. This will offend your beliefs. So, already we got you. Already, that says something. Will you take a step towards that path of critical thinking and explore these subjects and topics that I just communicated to you? These things are somewhat obvious if you know the biological processes. Or will you simply take the easy path, the centralized path, and shut it down? Now, back to the genetic discussion. With 16p11.2, there are implications with excitation and inhibition. That is, an imbalance between the two types of cellular functions, two cell types. All of this information today is a step up to properly cover the EI imbalance. 16p11.2 implicates cortical development, same as the artificial light mentioned previously. That has severe implications to our cortical development, just like the light sources I mentioned earlier, and the myelination. Brains and our biology aren't developing properly. (16p11.2) implicates that cortical development. But what is not known, it is unknown, what's causing the complications with these things that we're discussing today. Remember the artificial light exposure. Remember that tech light exposure. Remember the protein that I mentioned with melanopsin. And remember the circadian mismatch. Humans, modern humans, are essentially the only species with a circadian mismatch. It's wild. And remember the lack of aromatic protein activation. Remember these modern world problems. Remember the development of electricity and that power grid. And remember, autism came about as early as the late 1930s with Leo Kanner and Hans Asperger. Next is Cullin 3, or Cul3, CUL3. Cul3 is a gene that makes a protein called Cullin 3. Cul3 is highly associated with autism and other neurodevelopmental disorders. Now, you know how I feel about the word disorders. I don't really like it. In the critical stage of development, when we're young, Cul3 is a part of a pathway connecting 16p11.2 proteins to determine brain development. A Cul3 deficiency in prefrontal area implicates social interaction and sensory gating, or a way of determining sensations to perceptions. How we respond to physical phenomena in the environment. Remember, with autism, it is typically referred to as an abnormal response to our outside environment. Cul3 is associated with this. Also, Cul3 deficiency causes low functioning in NMDA, that learning receptor I mentioned a little bit earlier. Cul3 deficiency also implicates stereotypic behaviors in the striatum. And lastly, neuronal excitability, that crucial EI relationship. The whole reason we are exploring this upstream components of our biology. Next is SHANK3. SHANK3 is well studied in autism. SHANK3 is involved in tissues and brain. It involves synaptic connections, or how cells connect and speak to each other. SHANK3 also involves the dendritic and spine formation, so more of that. Specifically, SHANK3 encodes proteins for the excitatory synapses. Again, synapses are how cells connect and communicate. They move across the body, or they move across the brain. SHANK3 will be involved in the sensory processing challenges of autism. In scientific literature, SHANK3 knockout is shown to implicate neurotransmission in the striatum, whereby synaptic density were implicated. And behavior functioning shows repetitive behaviors, anxiety, and deficits in social interaction, and poor motor coordination. Does all of that sound familiar with autism? Something I have not mentioned yet is dyspraxia, which is seen in autism. It's basically poor motor coordination, especially at younger ages. In short, think about how cells talk to each other and areas of the brain connect. Next is Neurexin and Neuroligins, or Neuroligands. This is more synaptic development and functioning. Neurexins are presynaptic. Neuroligins, or ligands, are postsynaptic. The synaptic cleft is, remember, 10 nanometers. That is the space in between each synaptic area. Neuroligands work alongside glia cells. Remember the myelination part. A type of glia cell called astrocytes builds the myelin. It helps build the myelin. Myelination creates that highway of connection. It turns gravel roads into nicely paved highways. That's myelination. Remember the explanation about B-movie. Remember how things just come fast. Remember how efficiency is a key characteristic of autism if you understand your superpowers. If you align with your ability, that becomes a superpower. It is my goal that more people with autism and others around them, especially others around them, understand autism is a collection of superpowers and super deficits. Less worry about the super deficits. More focus on those awesome superpowers. Neuroligin 3 mutations enhance repetitive behaviors. Neuroligin 3 deletion impairs synaptic inhibition, so more EI imbalance, and those medium spiny neurons in the striatum. Remember the purpose of the spines. CNTNAP2, our old friend, is a Neurexin family member used in excitatory synapses formation. CNTNAP2 highly impacts metabolism in the medial prefrontal cortex. The medial prefrontal cortex has the highest resting metabolic rate. The medial prefrontal cortex is involved in decision making, memory integration, social cognition, understanding of the self, cognitive flexibility, excitatory and inhibition with medial prefrontal cortex transmission includes social behaviors and helps determine mood. As we wrap up today, I hope you understood a little bit about the genetic complications associated with autism. Further, what's more impressive is what these genetic mutations are causing. A lot with the synapses and cells, a lot with implications to our sensory processing, and that balance. This balance is going to be crucial of excitation and inhibition. You need that healthy balance. However, because there is an imbalance or potential imbalance, we will get into the superpowers and super deficits, the things that we can do and we can do well, and the things we can't do or we struggle to do. So be on the lookout for an episode about excitation and inhibition. Lastly, know and understand those implications with the light environment in our biology. If anything, you can improve your life by at least understanding this, at least making minor adjustments so your life can be better and healthier. If you would like to contact me, please do, for any types of questions, any types of comments, complaints, criticism, whatever the case. I would love to hear and have a conversation with you. You can email me at info.fromthespectrum@gmail.com. Lastly, thank you for listening to From the Spectrum podcast.